Welcome back to the Jungle Histology online training series. My name is Ken Longenecker, and this is the second of 9 modules designed to teach anybody who's interested how to perform the methods my colleagues and I use to do rapid reproductive analysis on reef fishes. In this module, I'm just going to present a brief overview of the techniques. Before I show you those techniques, I'd like to explain why we chose a histological or microscopic approach rather than a gross visual or macroscopic approach to analyzing fish gonads. The simple answer is that we've compared our results when we simply cut open the belly of a fish and took a look at the gonads versus when we took those gonads and did histological processing and looked at them under the microscope. It turns out that with a visual approach or macroscopic approach, we got it wrong about half the time when we analyzed a small snapper. Sometimes we couldn't even determine the sex of an individual. Sometimes we got both the sex and whether it was mature or not wrong. The upshot here is that we tended to overestimate the number of mature females and underestimate the number of mature males. We got very similar results for a larger snapper. Again, close to half the time we got it wrong. In this case, we did a very poor job of determining whether an individual was mature or not. The end result was the same. We overestimated the number of mature females and underestimated the number of mature males. Right now you may be thinking, well, maybe you and your colleagues are just really bad at the visual examination of these gonads. It turns out that others have produced similar results. Grandcourt et al. Worked on a sweet lips, did visual examination of gonads in a study in 2006, later went back and did a histological examination on the same population. It turns out that their visual examination underestimated the female size at maturity, or the female adult size, by 11% compared to their histological study. Vitale et al. Found the same sort of thing for cod. Now, getting female size at maturity or female adult size wrong has huge implications for fishery management. When Vitale et al. Looked at what those effects might be, it turned out that visual examination overestimated the spawning biomass of females by 35%. Another reason why we emphasize histology is that it's required to diagnose hermaphroditism or sex change in fishes. We've known for a long time that fish can change sex, and it turns out that many coral reef fishes are sex changers. Although you may suspect that a species is a sex changer, you need to do histology to confirm it. So the reasons we emphasize histology in the rapid reproductive analysis is to get accurate size at maturity for a species and to be sure whether a species is a sex changer. So now let's go to a video giving you a brief overview of the jungle histology or rapid reproductive analysis methods. Don't worry if you don't understand the steps. We're going to come back and look at each of the steps in a lot more detail in future modules. For now, we just want you to get a feel of what it is that you'll be doing. Record the date, measure the specimen, record its length, weigh the specimen, and record its weight. Record the specimen number on a tag consisting of a large part and a small part. Put that tag into a specimen bag and then remove the gonads from your specimen. Here you'll cut open the belly, start freeing the gonads from the connective tissue, and eventually remove the gonad. Put the gonad into the specimen bag along with its tag, record what you think the sex of the fish is, and then fix the gonads with Dietrich's fixative, and close up the bag. Wait at least 24 hours for that fixative to work, and remove the gonads and the tag from the bag. Weigh the whole gonad, record that weight, Now take the smaller part of that two-part tag and put it into one of the wells of a tissue culture plate. Then you'll remove a small subsample of the gonad, no more than 2 millimeters in any one dimension. Put that subsample along with its tag into the well and then add some alcohol to begin the dehydration process. This next step is only for females that look like they're ready to spawn. Take an approximately 1 centimeter cross section from one lobe of the ovary, weigh that sample, and record its weight. Take the sample and cut it in half. Add it to a container along with the larger portion of that two-part specimen tag. Add a solution called Gilson's fluid that's going to help liberate the eggs from that ovarian sample. Cap it off and store it away. Now we're going to go back to those small sections that were no more than 2 millimeters in one dimension. We'll dehydrate those by taking them through several changes of alcohol. Remove the alcohol, replace it with new alcohol, remove that change of alcohol, and replace that with fresh alcohol. Next we'll go through a process called infiltration. Remove the alcohol and add an infiltration solution. Remove that infiltration solution and replace it with new infiltration solution. And then remove that infiltration solution. Our next step is to embed those tissues into a small volume of solid plastic or a stub of plastic. We'll do that by readying some containers called beam capsules. Place the smaller tag into a beam capsule, then place the tissue into the beam capsule. And load those beam capsules with embedding solution. Close the beam capsules and put them into a desiccating chamber, which here is nothing more than a diver's dry box with some desiccant packets. Later, you'll take out the beam capsules and remove those stubs containing both your tissue section and the specimen label. Now you can see you'll be doing that with a single-edged razor blade, so use extreme caution so you don't hurt yourself. When the stub's out, you can just place it loose into the desiccating chamber again. Later, you'll take that stub out of the desiccating chamber, look at the number on the label, and record that number on a glass microscope slide. Prepare your slide to receive tissue section by placing 10 drops of water on the slide. Place the stub into the microtome, which is a piece of equipment that you'll use to make thin sections of the gonad. And begin making sections. When you have a good section, remove it from the microtome and float it on one of the water drops. Continue sectioning And do that until you've filled up all 10 of the water droplets on the slide. Then place those slides on a warm surface to allow the water droplets to dry. Next, you'll stain those slides. And rinse off the excess stain. And put those stained slides on the warm surface to dry again. Organize your stained slides in a slide box. And then you'll examine your slides on a microscope. Here you'll determine the sex and the reproductive state and look for evidence of sex change. And you'll record all that information on the data sheet. Now let's go back to those 1 centimeter cross sections of ovaries that you put into the Gilson's fluid. I'm repeating the video here to remind you where we stopped that process. The job of the Gilson's fluid is to liberate eggs from the ovaries, and it'll begin to work by itself, but you're going to help that process along by shaking it every day. Next day, shaking again the next day, shaking again the next day, shaking again the next day until it looks like all the eggs are free. To choose which of those samples to analyze, you'll go to your data sheets and choose the specimens that have reached spawning stage, as determined by histological examination. You'll dilute those samples to a constant volume, record that dilution volume, stir up those diluted eggs, and use this special pipette called a Stempel pipette to remove exactly 1 milliliter of those diluted eggs. Next, you'll count those eggs and record the number of eggs. You'll do that 10 times. Finally, nothing is more frightening than the prospect of doing all this work and then losing your data. So we make copies of the data, and we do that frequently. We take photographs of the data sheets themselves. We also enter the data into electronic spreadsheets, and this is very handy because you'll use those spreadsheets to do some data analysis later on. That concludes the jungle histology overview. Again, don't worry if you didn't understand each of the techniques. We'll come back and take a closer look at each of the steps in future modules.